I am very pleased to introduce Professor Wagner, that is our first speaker in this session. Professor Wagner was group leader of the Institute of Molecular Pathology in Vienna, Austria, a director for cancer vaccines from Boehringer. He was professor for pharmaceutical biology and biotechnology, as author of 20 patents and more than 400 publications in the field of protein, gene, and cell therapy. And so we are really having a top scientist that will bring to us the recent knowledge on chemical evolution of carriers for use in nanomedicine. Please, Professor, the floor.
Yeah, many thanks for the kind introduction. It's a great pleasure to be here in this great place, in this great society. When my talk refers to nanomedicine, you know, in a general term, this means application of nanotechnology for medical purposes. In the more narrow sense, it means developing nanoscaled agents for therapy, so-called nano-agents. And I only will give examples, and you will hear from my colleague afterwards the next thing. Now, nano-agents, one application is well known to you, it's gene therapy. And this is, I'm not going into details, just a list of now 5 approved gene therapies on the market. You know, gene therapy has had up and downs. And for these reasons, I pointed out the times when clinical studies started for each of the 5 and the time when it got approval. And you see, it's often more than 20 years. The first gene therapy, ADA's kit, it refers to children without immune system. They've got treated with adenodeaminase gene using a retroviral vector, and now this therapy is on market. Adenoviral vectors have been used with a lot of problems also, even death cases in some patients, and there is a market in cancer for them. Lipoprotein lipase, an AAV-based vector, it got approved for some lipid deficiencies. It's on market, the first one approved by EMEA, but it should say the disease is so rare that it gets market off again because just there is not sufficient use of it. On the other hand, oncolytic herpes simplex virus, which are viruses which replicate in tumors only and not in normal tissues. They have been approved for immunotherapy against melanoma recently, and I think we'll hear more by my colleague Daniel Schermann on a big revolution of chimeric antigen receptor T cells. These are gene-modified T cells to target tumor antigens, surface proteins, and Novartis just got this one for CD90 positive B-cell lymphoma on the market. Therefore, you can see within about 30 years and a lot of up and downs, finally technology has translated into something which is good for human society. And I mentioned these are all viral vectors, but I should say at the same time we have synthetic oligonucleotides like these here, which are also on the market. They are small chemically synthesized nucleic acids. And also this, thinking about that everything came within the last 25 years is quite amazing. And it's more, it's not only gene therapy and gene transfer. You may have heard of CRISPR-Cas9. There is novel genome editing methods, very precise cutting, at least in theory, I should emphasize, of the genome. There is siRNA and microRNA, and I touch this field slightly. And based on these nucleic acids, there are also novel biomarkers on the market. Therefore, gene therapy has moved into the field, I call it gene medicines, and a big implications in this way. Now, to introduce myself, how did I enter the field? I was interested in developing synthetic vectors, not viral vectors, which have already now proven successful in translation, but non-viral vectors, and especially nucleic acids that are complex with cationic polymers. And this is actually an old field because almost, more than 50 years now, the first cases have been established. At that time, they used RNA from viruses, and they used polymers to strongly enhance the transfection process. When I started in 1987, also 30 years ago now, we developed what we called receptor-targeted systems. And the notion has been all our cells on the surface have a specific coating with receptors, which makes them unique. Also, they make tumor cells unique, and therefore we can address receptors by active targeting. What we developed was we used transferrin, which is an iron transport protein. All our cells need iron, especially those which are replicating, and the transferrin is transporting iron, and it transports it to receptors on cells which have overexpressed this, like tumor cells. And we connected this with a synthetic polycation, polylysine, and we were very amazed to see that they form these donuts that you can see. And they are in direct comparison size with a natural adenovirus. And somehow when I saw this, and this was 1990, we said, okay, spontaneous formation of structures like viruses, this is just a coincidence with a natural evolution, that there's spontaneous self-organization, and we had the idea of generating artificial synthetic viruses. And as you see in a moment, endosomolytic artificial viruses. Because we attached actually these synthetic donuts with a real inactivated adenovirus, because we knew when these nanoparticles would enter the cell via transferrin receptor, they would be delivered into what is called endosomes. But normally, iron and transferrin go out of the cell again, and you would not have a productive process. In case of adenovirus, it enters cells, but it disrupts these endosomes. Therefore, we had an inactivated virus with no active genes. We had the gene in our artificial particles, and this way could very effectively deliver genes into cells by a synthetic process. And this was so effective even in primary cells, in primary tumors from patients, that it resulted in the very first polymer-based gene therapy trial on the world in 1994. It was a GMP-based, company-driven trial. I went to Boehringer Ingelheim, became production research and development manager. I was just not the doctor who applied this, but maybe everything else. And we had actually in end-stage melanoma patients, this was an interleukin-2-based cancer vaccine. We had complete responses, but we had a lot of no responses or only partial responses. And at that time, immunology was in its infancy, and now we have much better understanding about immune suppressive factor. And just now, there is clinical trials ongoing, like in Mainz and in other places, which give dramatic good effects, like in melanoma, combining the activation of the immune system with blocking the immune suppression by various means. Therefore, again, things take time. Now, what I learned from my company perspective at that time, before I went back to academia again, was if you want to do some pharmaceutical development under GMP, you have to fulfill certain rules. First of all, keep the system as simple as possible. But not always it is possible to keep it simple. Therefore, secondly, you have to make it precise. And the strategy we recently designed, and this is the main message of my whole talk, is design them as sequences and optimize carriers by, we call it chemical evolution. And I explain this in the next slide. First of all, why cannot we have it almost very simple? The reason is obvious. If you think about such a delivery process like in gene therapy, you have nanoparticles that have to be stably transporting the cargo in the blood systems. Then at the target cells, they have to attach in a specific way. They have, like I explained before, go endocytosis and endosomal escape. But now at this point, the same particles have to release the nucleic acids such as RNA, antisense, microRNA, or DNA. And please note, these particles at that point, they have to be stable, but just some minutes before they have to be dissociating, but some minutes before they have to be very stable. Therefore, these particles have to be dynamic. Of course, they are not intelligent, but they are bioresponsive. They cannot decide upon their own fate. They have to be programmed to undergo such a program. And viruses do this, and nature has shown us, and they can be generated in a precise form like natural viruses are. Now, we went further, and of course, viruses are built by natural evolution. And what is the key point of natural evolution? That everything is encoded in sequence. Think about yourself, think about all your proteins, all your nucleic acids, or everything you have, you are encoded by sequence. I don't say that you are programmed. I think we have our own will and we can change every second what we want. But all our materials are based on sequences. Now, what are the sequences based? Therefore, you need building blocks for the sequences. Nature has the nucleic acid and the amino acids. But we can use, at least the chemist, artificial building blocks. And especially those which we have found by whatever reason, which work well in such a delivery process. For example, and you have to assemble this chemical motif into sequences. I give you one example, which actually was the next speaker co-discovered and co-invented together with Jean-Paul Baer. This is a diamino acid motif. It's a proton sponge, and it has resulted just empirically in one of the most common transfection agents. We can take this information and build it in the unit, and so on. If we have defined sequences, we define them by using solid phase polymer synthesis. Solid phase synthesis we use is an important tool for the whole genome research. We have oligonucleotide synthesizers, peptide synthesizers. You can have chemical synthesizers for artificial sequences like we do. And what do? If you want to have an evolution process, you have to make systematic mutations or shufflings of domains. You have to screen them on a defined target, select candidates, again, fine-tune by a next round of selection until you get the structure which is most optimized for your aim. Now, in our case, a very talented PhD student, he developed solid phase polymer synthesis, which means you have a solid phase and you can put the various building blocks onto. You can wash away easily, you have no purification problems, and can get very good coupling degrees. And this way, you can make this, like here, artificial oligomers. And I should say, in that way, we mentioned it already today, there's a nanotoxicology issue. You can assemble sequences which have excellent biocompatibility. I give you a few examples, and they more or less just support my main message. For DNA delivery, the question has been, how long could the carrier be to be optimum? And we started with this nitrogen-containing artificial amino acid having 5 nitrogens, and we asked for DNA delivery, how does the size of the polymer correlate with gene transfer and toxicity? And what we actually looked at and what we found is that with 20 units having 100 nitrogen, which is quite a medium-sized molecule, you find a very good DNA compaction, and this way protection of the DNA. The highest gene transfer using a luciferase marker gene, actually better than the previously optimized polyacylanamine, and you find a low polymer cytotoxicity, far lower, about 10-fold lower than the classical agent, which also has been used actually even in clinical trials. Therefore, this is good to know, and by having solid phase synthesis, you can make this precise structure-activity relation. Classical polymers are very polydispersers. They have different blends and size ranges, and it's difficult to make a modification. Now, another example, we want to target them to specific tissue organs or tumors, so-called receptor ligands, and we can incorporate them. We can use chemical or also peptide-based sequences. This is one example where we targeted hepatoma and late prostate cancer, and they have the c-Met, which is the hepatocyte growth factor receptor, overexpressed, much more than normal tissues. If you incorporate a specific ligand for this, and you look for intercellular uptake of the DNA particles, you see by the red curves, this is fluorescence by flow cytometer, you see in about more than 100-fold enhanced uptake if you have this peptide for targeting, but not if you have, for example, alanine as a control amino acid. Now, this not only works in cell culture, if you properly package your DNA with this component, you can see it also works in a mouse system by systemic delivery. You can get a 50-fold higher gene expression in the tumor, but only if you have this targeting ligand, not if you have a control amino acid at the point. I should say the axis is here broken because otherwise it would be above the screen. And we then have used this not only for marker gene, but for therapeutic applications. For example, if you have a plasmid which is coding for sodium iodide symporter, this is a symporter that normally is present in the thyroid. It's important for the iodide uptake. If you now use this, but you deliver it into the tumor, in the control experiment where you don't use a functional gene, you see nothing in the tumor, but in the real group, you see here now the tumor glowing. This is using a diagnostic iodide isotope. And the same one is actually used by medical people in the clinics. And if you take a therapeutic iodine radioactive isotope, you get a tumor reducing effect. Now, instead of hepatoma, as we used here, of course, the concept can be generalized. It can be used using different targeting ligands and different tumors. And in case of glioma, we work together with the expert group from the Fudan University. And in that case, we use the peptide derived from interleukin-6, which can target the interleukin-6 receptor. Now, if you think about glioma, you have two barriers. First of all, you have the blood, and this is just an artificial model for the real situation, and you have so-called blood-brain barrier. It's a very tight endothelial layer which usually cannot be transferred. Now, with this particular active structure, you can actually see that there is transport across this tight barrier, and not if you have a control sequence. And in this model, you have the glioma below, which also have IL-6 receptor because it's an autocrine loop for them, and you can see that these are now transfected. Therefore, the particles can cross one layer of cells and can transfect the lower one. I should say glioma is the least dense blood-brain barrier because of the tumor inflammation. You have already some opening, but it's still one of the barriers for delivery. And the system has been used in real mice, tumor-bearing mice, and you see it did not really cure the mice, but it prolonged the survival of the mice better than temozolomide, which is a classical drug for glioblastoma. Which glioblastoma is really a terrible disease and there's no good therapy. This is a registered drug, and this simple system here already gives, very well competes with the classical drug. Now, DNA is a large molecule, and the main point of carrier is to package it in small nanoparticles. But there are small RNA molecule like siRNA. In that case, the aim has to be different. It means more protecting the siRNA. Therefore, a different cargo, a different carrier. And I would like to shortly introduce and explain the idea of RNA interference. You all know we have a genomic DNA. We heard already only 1% is transcribed and translated in the mRNA and translated into protein. We have about 25,000 encoding genes, but we have in addition 2,000 other genes which are microRNA genes. And they just produce a small piece of RNA which then gets incorporated into a so-called RISC complex. In general, you talk of this complex as active microRNA. And this microRNA is not expressed in every tissue. It's expressed in certain of your tissues. And some of this microRNA has an overlap with some of the mRNA, the messenger RNA, not every messenger RNA. Therefore, if in your cell you have a gene which happens to have a microRNA target sequence, and if in the same cell you have expressed, and if the same cell you have a microRNA that can bind to this mRNA, then you have a block of the RNA and you have no protein. Therefore, you have a very nice logic switch on regulating your genes on this post-transcriptional level, and of course you have transcription factor here that also regulates genes. Now, the interesting thing is these are very small molecules. You can introduce them into cells, and you can now manipulate your cells, like the tumor cells. You can introduce siRNA, then they shut off proteins artificially. You can introduce microRNA, they do the same, and you can introduce anti-microRNA. You can block the microRNA, you can turn on genes. Now, this RNA delivery, like I said, has different rules, and to show you one example, we were able to package it in a multifunctional nanoparticle, which a hydrodynamic radius of only about 3 nanometer, which means about 8 nanometer in length and less than 3 nanometer in diameter. And this contains all the necessary functions for entering the cell, like a ligand for uptake, like folate, like a peptide from a virus derived for endosomal escape, stabilizing groups which release the siRNA inside, and applying the system, quite simple, into a tumor. It retains by receptor interaction for like here up to 5 days. This is near infrared imaging. Without targeting ligand, it's lost after 1 day, or with free siRNA is lost within about 1 hour. Now, this system did not cure the tumors, but a small refinement did. I should say, first of all, when we used in therapeutic experiment, we introduce the siRNA against EG5, which is a kinesin spindle protein, it prevents the separation of the daughter chromosomes upon cell division. Therefore, what you now see here from these tumors under treatment, you see this double nuclei. This is when they could not separate, so-called aster formation. And it's very specific. You have the right siRNA. It would never happen if you take any control siRNA because it's a very specific process. And if cells can't divide, finally they go in apoptosis. Now, I said that with EG5 alone, there was no therapeutic effect or no convincing therapeutic effect, but a small change was now replacing folic acid with an analog, well-known analog, which is methotrexate. And it inhibits an important enzyme in the cell simultaneously with the gene transfer process. And if you took this, you see here now the application scheme, the tumors, only in this right tube, they shrink the tumors, and even long-term, half of the mice were completely cured. Therefore, it's a combination of a chemotherapy aspect and RNA interference aspect. I mentioned before that microRNA is a real remarkable novel topic in all areas of cell biology and also therapy. And we have been focusing on microRNA 200C. Why? Because this is a microRNA which breast cancer gets rid of when it gets aggressive. If you look at aggressive breast cancer, it has lost microRNA 200C. Benign breast cancer still has it. What is the function of microRNA? It can actually block the transfer into a migratory so-called mesenchymal stage. It can block cancer stem cell ness. Many of the cancers produce so-called cancer stem cells, which is difficult to treat. And they also block chemoresistance. They block chemoresistant genes. Therefore, with microRNA 200, you have less chemoresistance. Now we introduced such, and of course, I forgot, it blocks KRAS, which is an oncogene driving cancers into proliferation. Now, we wanted to reintroduce microRNA 200C into these cancer cells. In that case, we used lipopolymer structures, again, packaging siRNA, now in larger particles, which can be applied via the bloodstream. And in that case, we used epidermal growth factor receptor as a target because this aggressive breast cancer have this molecule on their surface. We use a peptide that binds this, and we could show that the presence of this targeting and, of course, the presence of the right 200C was essential for a biological effect. This, for example, is an inhibition of tumor migration, so-called Kretsch assay. When you apply this right material, tumors cells migrate much slower and don't fill in the window like in the control. And in that case, there are two controls. One is having the microRNA but no EGF receptor targeting, therefore it doesn't work. And the other one having all the good is EGF receptor targeting but having not the right microRNA. Therefore, both is important. You find reduced proliferation of these cells, and you find an enhanced chemoresistance against doxorubicin in this particular experiment. Therefore, it's a modulator also of these factors. Now, similar structures we analyzed in more detail, we made a library of many different lipo-oligomers combined with folate receptor targeting. And by the library screen, we found out that one was superior over the others. And we later found out it stabilizes the siRNA by some tyrosine residues. When you look at this in detail by, it was actually Professor Boga, by electron microscopy, you can see actually you label each siRNA with one gold nanoparticle of 10 nanometer. Therefore, you see here 2 nanoparticles entering by endocytosis into the cell. You can immunostain the folic acid receptor, and maybe you see that there is some red dots, which are the receptors, which are important for the uptake. We can follow with time. These are migrating into various organs. The organelles, I wanted to say, they are labeled with the fluorescent dye. And finally, you find them like here in the lysosome. This is work by Professor Kadoga in his lab. These particles, upon systemic delivery, accumulated in the tumor only if they had folate, not the non-targeted structures, and you will not be surprised by in that case we used again the EG5 siRNA, by the messenger RNA expression is now reduced by 2/3, and this is a lymphocytic leukemia tumor model which carries a folate receptor, and most importantly, clinical biochemistry was very clean. There were no side effects of this, or no significant side effects of this therapy. The same system has been applied for another targeting ligand. This is now again blood-brain barrier, glioma. Here there is AngioPep. This is a peptide targeting LDL receptor-related protein at the blood-brain barrier, and only if you had this peptide as part of your particle, now you get accumulation in this brain tumor. This is where this red dot is. There is also brain tumor in this group, but there is no targeting here, and only with this system you can actually silence a glioma gene, BAG3, again, like in the slide before, to about 2/3. And we know from other data that 2/3 is significant in vivo to have a significant therapeutic effect if you apply it more than one time. Now, technology is an important aspect. One point is, like in chemistry, you all know the famous click chemistry. Therefore, we have made libraries where you had an A site as part of these lipo-oligomer libraries. From nanoparticles, we can easily click them with several shields. And for example, this is one case where the nanoparticle has no shield, it accumulates in the liver. If you have polyethylene glycol or even more polysarcosine as a shield, you can appreciate that here now the whole animal, including the tips of the legs and so on, they are still containing large amount of this siRNA just by the shielding. And we have a lot of other opportunities now optimize these carriers, introducing disulfide bonds to reduce their toxicity, to make them really biodegradable after the job. And again, this shows a folate acid receptor targeting of the leukemia. Again, showing here 60% gene silencing, similar as I showed before. I won't stop before mentioning that this kind of chemical evolution can be used for other molecules. Proteins are now one of the biggest entities, certainly financially, in the pharmaceutical market. Proteins currently only address extracellular targets. We have done a screen for like green fluorescence protein with a library of many polymers, and what was found was here an oligomer which contains a cysteine disulfide bond and folate targeting and lipids. And they formed very interesting worm-like micelle structures where after 2 days you can see the cell is full of green fluorescence protein, really loaded. As you can see, 2 days, usually you have toxicities. They were very minor. But if you take a toxic protein like RNase A, showing here, and this is only this lower bar, you can completely or basically completely kill the tumor cells. This is a conjugate. If you leave away the disulfide bond here, you get a very mediocre effect. If you just take the RNase, there is no effect. This is no toxicity here. And if you have the polymer, it has no toxicity on its own. Now, libraries, sometimes the outcome gives you an idea what is going on, what you did not know before you started the empirical screening. And this is an interesting example. We were screening for nanobodies, which are tenfold smaller than normal antibodies for delivery. And the outcome was that the best oligomers made a cage around the molecules. They bound, and they made a disulfide formation after binding, and after cellular uptake in the cells, the disulfide bond removes, the cage opens again, and the protein is inside the cell, and you can actually use it for showing living cells under microscopy binding nanobodies to their target, for live cell screening. And going smaller and smaller, you can use it for small molecules in the design, but I would say medium small molecules, because one of the most interesting class is natural products. You all know Taxol, you know, these kind of agents. The rationale is twofold. You can have tumor targeting for reducing side effects and having a better pharmacological profile. And there is a stealth liposome called Doxil, which strongly reduces the cardiotoxicity of doxorubicin. And paclitaxel, Taxol has been formulated as albumin to have these abraxane nanoparticles. But there's a second point. Some of these medium-sized molecules don't go as effective into cells as they might do. Therefore, having a better intercellular cytosolic delivery, and this shows you two recent examples, pre-tubulizing, very highly effective tubulin inhibitor, 1 nanomolar IC50, very potent drug, having the formulation after a screen, you can see 0.1 nanomolar. We have even tenfold enhanced the potency. We are now 0.1 nanomolar IC50 with these molecules. And a similar drug, which is not so, it's an active inhibitor, not so potent, at least not in this particular cell type. You see, this is a drug without help. Taking the same drug with the carriers, we are now these curves, and we have again both an increased IC50, but 100% or almost 100% cell killing here. Now, where is the journey going on? If you say chemical evolution, we have to do the evolution in the real situation. We cannot do it in human. We cannot even do classical screening like in cell culture in mice. But what is the concept? That we would not screen single nanoparticles, but we use, actually in our lab, barcode siRNA. Therefore, each nanoparticle contains a different code, but has different siRNA. Therefore, we can measure in the tumor the siRNA. This is one example of single experiments, and then optimize the delivery. I should say, like always, innovation goes in parallel, and very recently there was a nice paper by Dahlman in PNAS for DNA barcode nanoparticles. This is a group from Boston, and they were faster than we, but it's just confirming that this kind of barcode concept is a good idea for optimization. Therefore, my summary, the aim is to have a higher potency and safety of nanomedicines and medicine in general by targeting and better intercellular delivery. Possible solutions are, as I presented, a bio-inspired chemical evolution like nature has done, but on an artificial synthetic background. I would not stop without giving you the caveat. These are not easy tasks still. Warren Shan has shown only 0.7% of nanoparticles have reached tumors by searching literature. And I also want to say it's not the only way to use biochemical evolution. Physical targeting is one of the points, like the next speaker will talk. Pharmacy on the microchip by microelectromechanical systems is another really interesting development of having controls, pulsed delivery of pharmaceutical drugs would be helpful. And I stop by acknowledging the group and thanking for your attention.
For two very short questions, please. You are there? No? Okay, please.
Yes. Can you hear me? Yes.
nature uses protamine and histones to compact the DNA. Have we thought of using modified protamines or histones instead of polyethylenamine? Because one of the issues with polyethylenamine is that how you biodegrade this and how you get rid of it. It needs to go out of the cell, and then it needs to be excreted. And secondly, these polyethylenamine derivatives are very pH sensitive because the degree of protonation depends on the pH, and they are not sequentially protonated.
No. Yeah, excellent comments. I just can agree. Do you think the non-degradability of PI, if we have sequences like we have, where we have active disulfide building blocks, inside the cells we have reducing agent and they will be degraded. And there's a recent issue in molecular therapy together with Professor Mogimi. We have actually shown already these novel structures have less effects than like PI. But your point are perfect, yeah.
So if there is no more questions, thank you again, Professor Wagner, for your talk.